Okay, great. Let me just see if I can get a pointer. Ah, okay, perfect. Yes, thank you very much for joining me. Actually, I'm not going to talk about nervous system regeneration so much, but just want to share with you the main concepts of regeneration in vertebrates and what we can learn from studying different regenerative and also non-regenerative model organisms. Oops, and now I cannot move. I guess you might have heard it, but if not, I just want to um, mention this, that regenerative ability is quite unevenly distributed among animals, and especially among vertebrates. We can see quite dramatic changes when we look at different vertebrates and their regenerative abilities. So for example, fish and amphibians are rather good at regenerating, and I will go into more detail of this later. And then when we look at mammals, including us, we are rather poor in regeneration. But there are always exceptions. So even though it is largely said that mammals are poor regenerators, over the past years, especially the so-called spiny mouse has really rise as a model system because it is a mammal that can regenerate. And this is quite an extreme study. You can see that this mouse is losing parts of its skin. This is not that people have ripped off the skin of this mouse, but it's actually this mouse can also shed its skin when it's attacked by predators. And you can see here these large skin wounds, which can regenerate within only 30 days. And there's new hair regrowing, which is very much in contrast to what happens in our skin. When we lose parts of it, there is mostly scar tissue forming. But even though there is the spiny mouse, I want to highlight that fish and salamanders are really the champions among vertebrates when it comes to regenerative abilities. And there are two model systems that are widely studied. One is the zebrafish, Danio, and then the axolotl, which is a salamander. They can regenerate many organs, including hearts, brains, retinas, and then limbs and fins, which I will go into a bit later. So basically, I want to go through these three big that regeneration biologists are interested in, and then tell you what we have learned by studying salamanders mostly about these questions. So the first question is, what is the source of the regenerated cells? Where do they come from? And then secondly, how is this regeneration process after an injury actually started? What kicks off this process? What activates these source cells that make new cells? And lastly, one big question, which is also really important, is how are the correct cells regenerated to really ensure that what has been lost is regenerated and structures are appropriately recovered. So I want to start with limb regeneration in salamanders, and this goes back to this Italian philosopher and scientist and Lazzaro Spallanzani, who was the first one to realize that salamanders can regenerate. And this is a letter that he wrote to a friend and colleague of his, where he basically drew how salamanders, after amputation of the tail, can regenerate new tail tissue. And this was in 1766. And then a bit later, his colleague Charles Bonnet also studied salamanders and looked at their limbs, and he amputated the limbs of these salamanders, and you can see it here in these drawings, how limb regeneration can occur in salamanders. So we knew this quite long ago, that salamanders can regenerate lost appendages, and this is here the process that a salamander limb goes through when it's amputated. So the first thing that happens really quickly within the first day is that the wound is healing, and there's this specialized wound epidermis that is forming above this injury. And then one of the really crucial steps is the formation of this so-called blastema, which is a pool of progenitor cells, and I will explain this a bit more later. And later, this blastema will proliferate, grow out, and then eventually patterning will occur. And you can see here these digits are reforming so that in the end, this limb looks really well regenerated. Now, to the first question, what is the source of the regenerated cells? This was something that people were really studying a long time ago in the salamander and in the limb. And this is actually a very big question, because when we look at the cell types that exist in the limb, there's a large variety. There's skin, there's muscle, There's the skeleton, there are nerves, blood vessels, lymph vessels, and blood cells. So who is actually regenerating what? And there are different ways to regenerate. I'm just highlighting the two most common ones here. One is the dedifferentiation of a committed cell. So you have a cell type that is differentiated. Let's say it's a muscle cell. And then upon an injury, for example, the cell could transition into a more progenitor-like state. And then after that, regenerate various cell types. The other option that we could think of is the expansion of a pre-existing progenitor cell. So this means that in the tissue already there's a progenitor that has the ability to regenerate certain cell types, and this progenitor just needs to be activated and then can regenerate what has been lost. Now in salamanders, this has been really studied over the last 20 years, I would say. You can see here, this is the first transgenic axolotl that has been made. That was actually now 20 years ago, where this GFP positive axolotl was made. And with these animals, people could actually start to figure out what cell types contribute to regeneration and what other cell types do they contribute to. And this was done by tissue grafting, which can be really nicely done in salamanders. So one can take a piece of this GFP positive salamander, transplant this tissue to a non-transgenic salamander, and then in the case of the limb, do an amputation, and then, as you see here, track the fluorescent cells. And this can be done in different ways. So up here you see this is from a mature salamander to a mature salamander, but they can also be like embryonic tissue graftings, which can then allow to label certain lineages of cells. So what have people found? There's one example that I want to show here, which is that it has been found that muscle makes muscle in limb regeneration. So here they use these embryonic transplantations where they use presemitic mesoderm cells and they transplant from this GFP positive animal to a wild type animal. And then because it's presemitic mesoderm, only muscles of the limb will be labeled. And you can see this here. This is before the amputation. There's this green muscle. And then after amputation and following this limb throughout its regeneration process, you can already see it here. This looks very similar to what has been there before. And then by using stainings, for example, for satellite cell markers, which are the muscle stem cells, and also muscle markers, it was found that muscle just makes muscle. So it looks like in the case of muscle, it's just the expansion of a pre-existing progenitor. You have muscle stem cells, they will start to proliferate and they will generate muscle cells. And here's a summary of what has been found in this very, very important study that was published in 2009, is that most cells actually keep a memory of their tissue origin. So I talked about muscle, but this has also been shown for dermal cells, for cartilage, Schwann cells, epidermal cells. And then there was one cell type that was kind of missing and interesting, which were the fibroblasts. So they were not tracked in this study, and it was unclear what fibroblasts actually contribute to in limb regeneration. Now we fast forward a bit. This was in 2018, where single-cell profiling had made it into molecular biology and also into the axolotl. And in this study, single-cell profiling of fibroblasts was performed. And here, a time course of limb regeneration was performed from 3 days post-amputation until 18 days post-amputation, and always fibroblasts and their descendants were sequenced. And as a comparison, people also looked at the developing limb to really understand whether these fibroblasts might actually activate transcriptional programs that are similar to what happens in limb development, or they just do something that is very different. And now I just want to show you or highlight this graph here, where you can see a comparison of these fibroblasts and their descendants to the limb buds or the developmental cells. And what you can see here is that the higher this bar goes, the more similar these fibroblasts come to these limb developmental cells. And what we learned here is that 11 days post-injury, these fibroblasts become really similar in their transcriptome to the limb developmental cells. So this was the first evidence that these fibroblasts might actually go back in time and de-differentiate to a more embryonic limb bud-like cell type. And then the other experiment that was performed was more to look at whether they also now possess functional similarity to limb bud cells. And this is a test for whether these fibroblasts can now make bone cells in regeneration. So here, fibroblasts that are marked with M. cherry have been transplanted And what you can see here by co-labeling with SOX9, which is a bone marker, these fibroblasts now can contribute to bone. So in this case, we learned that fibroblasts dedifferentiate, and both on a transcriptional and a functional level, they become more similar to lymphoid cells. So this is now more the second option that I highlighted, where we see dedifferentiation of a committed cell, the fibroblast, to more progenitor-like cells, which are the blastema cells mostly, and then they can differentiate into different cell types, such as cartilage, bone, and also other non-skeletal connective tissue types. These fibroblasts are really important, and this becomes very clear when we look at other species. For example, post-metamorphic frogs are not able to regenerate their limbs after amputation. You can see here a frog limb before amputation and then a time course of the regeneration. And I guess it's really clear that this structure that comes out at the end is not very similar to the original limb. This is the so-called spike that forms. So the question here was, what's going on? Are these fibroblasts maybe different in the frog, and what occurs with them? So in this study, people did a very similar thing to what had been done before in the axolotl, where fibroblasts of the frog have been analyzed. And again, first they looked at transcriptional similarity and whether these fibroblasts also perform dedifferentiation and become more similar to lymphoid cells. And now here on the left side, you see the plots for the frog, and on the right side, the plots for the axolotl. And as in the previous study, you can see that the axolotl fibroblasts, they become transcriptionally really similar to the lymphoid cells, which are up here. And the frog cells, even though they can make it a bit, they don't really cross this line and don't become transcriptionally similar or as similar as the axolotl fibroblasts. And then again, on a functional level, also these transplantations have been performed, and it has been found that these fibroblasts are not able to generate new bone. So in contrast to the axolotl, post-metamorphic frog fibroblasts do not dedifferentiate sufficiently, which might be really the key of why this frog limb cannot regenerate appropriately. So this was about the dedifferentiation, and now I just want to highlight one other example of expansion of a pre-existing progenitor, and this is what occurs when we look at the nervous system. So for example, in the axolotl brain, we have stem cells that are present both in the homeostatic and also in the injured brain. And you can see here, this is an EDU pulse chase experiment where S-phase cells, so proliferating cells, have been labeled, and all these cells that are sitting here in these layers are neurons. You can see here that a homeostatic brain has proliferating stem cells and they generate some neurons. And then the thing that happens after injury is that these stem cells get activated to proliferate more and generate more neurons. So in this case, we really have pre-existing stem cells that need to be activated to proliferate more than they do in homeostasis. And again, this is really in contrast to non-regenerative organisms such as the mouse, for example, where after a brain injury, you see these large gaps that really remain after injury, and there's no new neurons made, in contrast to the axolotl, where you can see also large injuries can be performed, and this animal is really able to regenerate these injuries. So to summarize this part, when we look at the origin of regeneration in the limb of the axolotl, we learned that most cells keep a memory of their tissue origin during regeneration. And it's really the fibroblasts that do something rather different. They dedifferentiate to a more embryonic lymphoid-like state, and they can also regenerate then, for example, the bone. From these comparisons to the frog, we have learned that fibroblast dedifferentiation is really a key determinant of successful limb regeneration. And then I just highlighted to you the axolotl brain, which uses a bit of a different strategy, where we have these pre-existing stem cells that can regenerate what has been lost. So now to the second question. How is this regeneration process actually started? This was a question that interested many people. And in this study in 2016, people did an unbiased screen where they cloned 25,000 axolotl genes, transfected them into human cells, and then cultured the supernatant containing axolotl proteins. and put these proteins onto salamander cells to see if they can induce proliferation. And from all of these 25,000 genes, there was one gene that really stood out, and this was axolotl MLP, Marx-like protein, which could really induce these salamander cells to proliferate. Interestingly, axolotl MLP is also expressed after a tail injury and also a limb injury. And it's actually really early on. You see already at 12 hours after the injury, there's a peak in tail amputations, and 1 day after injury, there's a peak in limb amputations. And then importantly, when you provide this axolotl Marx-like protein to, for example, the tail, cells will start to enter the S phase. So you can see this here. On the left-hand side, you have purified axolotl MLP, and on the other side, this is just the flow-through of the column, which should not contain MLP. And I hope you can see here, there are these BRD positive cells, which mark S phase entry. And this is also quantified here, that actually, for example, in the spinal cord, MLP is able to induce proliferation, but also in other tissues such as the epidermis and even PAX7 positive satellite cells. Now, really important was also to understand whether this might be necessary for regeneration. And this experiment here was done with morpholinos, which should block the translation of the axolotl MLP. So on the left-hand side, you see this is a control where, again, the flow-through or the purified protein has been injected, and a control morpholino is provided. And you can see there's almost no difference between these two tail regeneration paradigms. But now, when you block axolotl MLP and you only provide the flow-through, you inhibit regeneration. But then once you provide again axolotl MLP, this can rescue this regeneration defect. So from this, we really learned axolotl MLP is a protein that gets expressed really early after injury and is necessary and also sufficient for appendage regeneration. Then, of course, there's a question, which cell types provide actually molecules? There's MLP, which we know now might be secreted from the epidermis and from nerves, but there might be also other cell types involved in initiating regeneration. And there was one study that was really important for the field, which showed that myeloid cells, such as macrophages and other immune cells, accumulate really early after limb amputation. You can see this here in these images already at 1 day post-injury. These dark cells that are stained here, these are myeloid cells that accumulate at the injury site, and you can see how they are more and more invading this injured blastema. This is also quantified here, that these are different cell types. They're myeloid cells in general, then also granulocytes, and then here different marker stainings have been performed to look at monocytes and macrophages. Basically, the picture is that, yeah, myeloid cells accumulate in this regenerating blastema. Now, the question was, is that actually good or bad or necessary for regeneration? And the strategy to test whether this macrophage engagement might be important for limb regeneration is using these clodronate liposomes. These are liposomes that get phagocytosed by macrophages, and then once they are coupled with the lysosome, this clodronate is released and leads to apoptosis of the cells. So basically, you can just inject these liposomes into limbs, macrophages will eat them up, and they will die. And by this, we can deplete macrophages from the system and understand whether they're important for regeneration. And this was a really dramatic effect. So when these macrophages are depleted in the early phases of limb regeneration, you can see up here, this is a PBS control where these liposomes don't contain this clodronate, limb regeneration occurs. But then once clodronate liposomes are provided to the system, limb regeneration is blocked completely. And it's really important to see that this is really due to this early engagement of the macrophages, because when these liposomes are injected later, when regeneration has already started, you can see that the limb is regenerating, even though it's a bit smaller than in the controls. And with this study and many other studies, the field has actually learned that inflammation is rather pro-regenerative also in other systems. For example, now we know in the salamander limb and also in the heart that inflammation is really necessary for initiating regeneration. The zebrafish field has also found this in the fin, the heart, the retina, the brain, and the spinal cord, and even now in the spiny mouse skin, and also in digit tips of the mouse, people have seen that inflammation is rather pro-regenerative. Now there's one other cell type that is really crucial for regeneration, and these are nerves. And this goes back really quite some time ago to 1823, when Tweedy Todd wrote that if one cuts the nerve after the healing of the stump of a limb, then regeneration is retarded or prevented. And people have then looked at this much more in salamanders, in this case of a newt, where when you cut the nerve and then also amputate the limb, regeneration of the limb is completely blocked. And again, this is a really depending on the timing of this nerve transaction. So Jeremy Brox, he had this nice paper where he basically showed in these drawings how when you do early nerve transactions, regeneration is inhibited, but once regeneration has already started and the nerve is severed, the limb is not really dependent on the nerve anymore. So the question that the field had was, of course, what does the nerve provide, or why are nerves actually important? And there's important things that people have found, which is that it does not depend on what type of nerve is there. It's really all nerve fibers that contribute to this regeneration process. And it's rather the amount of nerves that are present that trigger regeneration. So this was the so-called neurotrophic hypothesis that Markus Singer postulated, where he already speculated there must be factors that are secreted by the nerves that induce regeneration. And over the past years, many different factors have been found, such as substance P, insulin, transferrin, nerve growth factor, FGFs, BMPs, neuregulin, and others. And there's one factor that I want to highlight here, which is this anterior gradient protein. This was one of the first factors where people really found that it is locally expressed on nerves after the injury. So you see here, this is the sheath of the nerve, and this is the wound epidermis in a 5-day blastema of a newt. And in green, there's this anterior gradient protein stained. And also here in a cross-section of a nerve, you can also see here in green the expression of this protein. Now, what was really exciting was that in the study, it could be shown that actually this newt anterior gradient protein is sufficient to regenerate when a limb is denervated and amputated. So in this experiment, again, the nerve was cut, the limb was amputated, and then in half of the animals, a control vector had been electroporated into the limb, or a vector containing this anterior gradient protein had been regenerated. And you can see it here in these pictures and below quantified. In the vector control, 0 out of 24 animals regenerated the limb. So basically, limb regeneration was blocked completely. And then when this anterior gradient protein was provided, 15 out of 29 animals could regenerate a limb and even form the digits. And again, this nerve dependency is a concept that has been now looked at in other systems and other organs. We know from the axolotl tail, the zebrafish fin, the mouse digit tip, and also skin wounds in the mouse that nerves are really important because they secrete pro-regenerative factors. So now I want to move into the third part, and this is the question of how are the correct cell types regenerated. I showed you this picture in the beginning where we looked at the regeneration time course, and what I didn't mention there is that this was an upper arm amputation. And I hope what you can see here is that indeed the lower arm and the hand are regenerated after this amputation. Now in this paper, there was also another amputation, which is at the lower arm level. And what you can see here is that also a blastema forms, digits reform, but it's only the lower arm and the hand. So only the pieces that are missing that are regenerated. Oops, I cannot move my slide anymore. Yes. So the question is, how does this work? How does the system know what is missing, and where is positional memory, if there is any, encoded? So now we know that positional memory exists in different axes. There's, for example, the proximal distal axis, which goes from the shoulder to the fingers. There's an anterior axis, which goes from the thumb to the little finger. And there's also a dorsal-ventral axis, which is back of the hand to the palm level. And really the first question that arose in the field was which cell types might contain positional memory. And this was a very, very nice experiment in this paper where they used, again, these GFP positive salamanders. In this case, there was only connective tissue labeled. And here they do an upper arm amputation in two animals, a white animal and a transgenic animal. And now they transplant this upper arm blastema, which contains GFP-positive connective tissue, to this white host and see what is regenerated. And what I hope you can see here in this picture is that the upper arm, the lower arm, and the hand are GFP positive. Now, when the same experiment is done where the donor is now a wrist blastema, what I hope you can appreciate here is that now only the wrist is GFP positive. So from this, we learned that the fibroblasts might be actually really the cell types that harbor this positional memory. And a good control here was that there was another transgenic salamander which labeled only the muscles. And when you now do this experiment of transplanting a wrist blastema to this upper arm stump, you can see that the muscles, they don't really care. They just reform upper arm, lower arm, and hand muscles. So again, it's really the fibroblasts that are very important for regeneration. And I just want to go into some of the concepts that we learned over the years about what these different positional information sites mean and how important they are for regeneration. So I want to talk here about anterior-posterior positional information. And we learned over the years that this is really critical for correct regeneration. What can be done in salamanders is that posterior and anterior sides of the limb can be flipped and stitched together. So people have generated arms that have anterior and posterior sides, but also posterior or anterior only arms. And what you can see here is happening is that limb regeneration is basically really defective. So on the left side, you have a control limb, which has these 5 digits. If you have these double posterior limbs, they form incorrect digit numbers, and also double anterior limbs also form incorrect numbers. They form rather spikes, which is very reminiscent of what happens in the frog. Anterior-posterior memory is also really important for inducing limbs. And there's this amazing assay where you can use this anterior-posterior discontinuity and the nerve, which is, as I told you before, really important for regeneration, to generate extra limbs. So this is the so-called accessory limb model, where one generates a wound in the skin. One has to deviate a nerve to this wound and then takes a piece of skin from the opposing identity. So in this case, posterior to this anterior wound site. And what you can see here in this drawing is an accessory limb. And here you can see how this actually looks like when this is done, in this case, in a nude or in an axolotl. So again, these anterior-posterior sides coming together is really important for forming a limb. So of course the question is like, what is the basis of that? And the Tanaka lab has published a paper this year which really went into what is the molecular basis of positional information. And now we know that, at least from the anterior and posterior side, there are genes which continue to be expressed in the mature tissue that had been there in development, and they just continue to be expressed. So in this case, what has been done is that connective tissue cells, so fibroblasts from either anterior or posterior side, were profiled by RNA sequencing. And there was one gene in the posterior side that stood out, which was HAN2. You can see here how HAN2 is expressed in the developing limb, but then also continues to be expressed in the mature limb. And then again, after an amputation, this gene gets really upregulated. Now, what was found here in this study is that HAN2 is really necessary for posterior limb identity. And this was again using this transplantation and accessory limb model assay, where now in this case, on the anterior side, a wound is made, a nerve is moved to this wound, and then posterior skin is transplanted to the site. And as I explained before, in a normal limb, if you use anterior and posterior skin and bring them together and have the nerve, an accessory limb will form. And this is what happens in a control. But now if you use the posterior skin of a HAN2 crispant, you can see that no accessory limb can be formed, which tells you that really HAN2 might instruct some positional identity, posterior positional identity in this, this part. And then the converse experiment is really exciting, which showed that HAN2 is really sufficient for inducing posterior limb identity. So what has been done here is again using this accessory limb model, but now it's an anterior piece of skin where HEN2 was overexpressed. So normally it's not expressed there, and now with forced expression, anterior skin expresses HEN2. And in this case, you see that skin with strong HEN2 expression could form this accessory limb, while again, in the contrast, when there's only weak or no HEN2 expression, there's no accessory limb forming. So to summarize this part, I showed you that we know now that wound epidermis-derived factors, nerve-derived factors, and also immune cells are really important initiators of regeneration in limb regeneration, but also in many other tissues and many other species. The regenerating blastema cells of the salamander limb harbors positional identity, and it's mostly these fibroblast-derived cells that are really harboring positional identity. And this AP memory is retained in mature cells by continued expression of some developmental genes, such as, for example, HEN2, which we now know is really necessary and sufficient to induce this posterior limb identity. So now in the last part, I want to move a bit to mammalian systems. I told you in the beginning that mammals, including us, are rather poor regenerators. And I guess you might know that mammalian limbs, when they are amputated, cannot regenerate. But there's one model system where we can study mammalian almost limb regeneration, and this is the digitip. And in this model, there are actually two paradigms. One's where an amputation is done in the phalange 3 and the nail is left intact, and this leads to regeneration of the digitip. And then there's the other amputation, which is a bit more proximal in the phalange 2, where then fibrosis occurs, a scar is formed, and no regeneration occurs. And this is a really nice model that people have used to compare regeneration versus fibrotic processes. And by now we know that the nail is really important because it contains stem cells that provide signaling. There's also really important signaling from the epidermis and again also from the nerves that induces this regeneration. These cells that are formed here in this injury site form a structure that really resembles the blastema in salamanders, but these cells are rather lineage restricted. And one really important factor that I will talk about in a minute or so is that there is soft extracellular matrix deposited in this injury site. Now, in contrast, when we do this more proximal amputation, where fibrosis occurs and no regeneration occurs, we know that the nail has been amputated, so there is no nail stem cell signaling center. There are more myofibroblasts that appear that will form the scar, and also the extracellular matrix has a very different composition. It's much more stiff. And this brings me now to the last part, which is, could we actually induce regeneration in these non-regenerative amputations? And this is a study from this year, from the lab of Michaela Storer, where they looked at the extracellular matrix. And I mentioned this before. So what they found here is that in these regenerative amputations, the extracellular matrix is much more soft, and there's a lot of hyaluronic acid and also linker proteins that link hyaluronic acid together present. So you see this down here in the staining. This is the regeneration paradigm, and up here is the non-regeneration paradigm, where this linker protein, HAP-LN1, is really strongly expressed. And here, using atomic force microscopy, they also saw that really in these non-regenerative amputations, the matrix is much more stiff than in the regenerative amputations. So now they asked again for necessity, and they could deplete hyaluronic acid from the whole system of the mouse using a special diet. And you can see here now, when you do these normally regenerative amputations in a control, the digit regenerates, but when hyaluronic acid is depleted, regeneration is blocked. This is because cells proliferate much less. And now what also happens is that the stiffness of this amputation is much more increased than in the controls, and looks much more like in the non-regenerative scenarios. And lastly, what was really exciting in this paper is that they now asked, okay, can we now use what we learned about hyaluronic acid and this more soft matrix to induce regeneration in these normally non-regenerative amputations? So they used this linker protein, HAP-LN1, and expressed this in fibroblasts and then transplanted them into digits, which were then amputated at this phalange 2 level, where normally there is no regeneration. And you can see this here in the control. In red is newly formed bone. There's a bit of bone forming in the control, but then when you overexpress this linker protein that can link hyaluronic acid, you see much more bone regeneration. And also this is seen here in stainings for RUNX2, which is a bone marker that, again, in the control, there's hardly any bone regenerated, and there's quite some amount of bone that gets regenerated when this HAP-LN1 is overexpressed. So what I showed you here is that mammals, also including us, actually can regenerate digits, but this is restricted to more distal amputations, and the nail is a really critical signaling center that needs to be intact for this regeneration to occur. There's a blastema-like structure that forms after injury, and these cells that are in the blastema, they are mostly lineage restricted. And lastly, I showed you this new study where people have seen that hyaluronic acid and soft ECM is really important for digit repair and can help to induce some regeneration after these non-regenerative amputations. So with this, I'm at the end, and if you have any questions, I would be very happy to answer them.